Good afternoon. Like Kamal said, my name is Robert Smith. I am the Terrestrial Restoration Resiliency Coordinator for SLELO PRISM, which, as Kamal said, is a very long name for St. Lawrence Eastern Lake Ontario Partnership for Regional Invasive Species Management, or SLELO PRISM, which is a lot easier to say. I'm going to go over a lot of stuff. In fact, more things than I've ever gone over when it comes to emerald ash borer. I know people have various levels of understanding and experience with emerald ash borer. I didn't come in assuming that everybody had, I came assuming that they had no experience, because there might be some people in here that have no experience with it. So I'll go over the general information about emerald ash borer, the various management strategies that are out there to control emerald ash borer, the biocontrol release program here at Rice Creek Field Station and as a whole across the country, and which we've done for the last 2 years, and the establishment surveys, which we're going to do this year and next year. So I'm going to go over all of that. We'll see how long that takes. Hopefully, I'll keep it within our time. All right. All right, for those that are not overly familiar with SLELO PRISM, I already told you what it stands for, but we're one of 8 prisms you can see on here, right here along the lake next to the eastern side of Lake Ontario, that encompasses 5 counties. So Oswego, Oneida, Lewis, Jefferson, and St. Lawrence County. So quite the big region. There's 7 other prisms. And the PRISM system was established by the DEC. And what the DEC does is it contracts out the managing of the prisms to different organizations. So SLELO PRISM and the Adirondack PRISM are both contracts between the DEC and the Nature Conservancy, which is my parent organization. So what do? We assist with the management of invasive species in those 5 counties. And this is done directly by us in what we call priority conservation areas, or areas of high ecological importance, and indirectly through partnering with other organizations. And we have a lot of other partner organizations. And what they do is they manage the invasives on their lands. And then additionally, we educate the public who manage. Oh, thank you. Additionally, we educate the public about invasive species, who in turn manage their own private properties and then report sightings of invasive species throughout the region. So it's a layered system that, because there's no way SLELO PRISM, which is a staff of 4, can do all this, because we literally have more counties responsible for it than we have people on the staff. But with our partners and the public, we're able to reduce some of these invasive species and prevent some of the spread of other invasive species. All right, emerald ash borer. Well, emerald ash borer is a emerald colored wood boring beetle. It's from Asia, and it kills all species of North American ash trees. No exceptions. And any of like the cultivar street tree versions of it, they also get hit there and killed by emerald ash borer. So we have no exceptions on that. It was first detected in Michigan in 2002. And probably arrived on solid wood packing material carried in by cargo ships or airplanes. The adults are about a half an inch long, they said, with green wing covers, and then they have a coppery colored abdomen. The adults only live about 3 weeks, and they just eat leaves, and not to the degree that it causes any harm whatsoever. So the adults are not the concern when it comes to emerald ash borer. They just nibble on a few leaves during that 3 weeks. It doesn't really cause any harm. What they do, though, is adults lay eggs in the bark in the crevices of the ash trees. And when the eggs hatch, the larvae will burrow into the ash tree where they feed. And it's the feeding within the ash tree that damages the tree. The larvae are white in color. They're about 1 inch in size, and they have bell-shaped things that you can kind of see there. They have bell-shaped segmented bodies. And just a little FYI that I found out while I was preparing for this, there happens to be one species that's from Asia, the Manchurian ash, that does happen to be resistant to EAB. For its worth, but no natives are. All right, new infestations are difficult there to detect because you won't see the outside, the damage to the tree for the first couple years. When the signs and symptoms of emerald ash borer appear, they include the S-shaped markings you see down here at the bottom right. That's on the inner bark and the phloem. And you can only see this if you debark the tree. So you won't see this out there. The other possibility is that the bark is coming off a dead tree, which I actually did see out here last year. But otherwise, you're not going to see that. D-shaped exit holes, those are formed when they emerge, the adults emerge out of the bark trunk of the tree. Crown thinning, which is also known as dieback. This is a result of the dying of the branches. So when the branches are dying, the foliage will start to turn yellow, wilt, and then it'll fall off the tree, leaving the branches bare. So then you start seeing thinning in the crown of the tree. Epicormic shoots, you can see a good picture there, the epicormic shoots. They're sprouts that they're like clumps of sprouts you can see growing off the trunk there, usually just along part of the trunk that normally there wouldn't be no branches on a healthy tree. You'll see these sprouts coming up out of the trunk. And that's really a response the tree has to the stress of the presence of the emerald ash borer feeding. Woodpecker damage, also known as blonding, is areas of the bark where the woodpeckers, which are trying to get to the larvae, they're pecking away at the bark, trying to get to that larvae. So some of the bark falls off, and the inner bark is lighter, so that's why they call it blonding. And that's around here that you'll see that for sure. And lastly, vertical bark splits. Those occur like over the bark area where the larval galleries are present. Impacts. As mentioned before, the larvae of the emerald ash borer burrows tunnels and feeds on the cambium layer. This feeding cuts off the nutrient supply to the infested tree, eventually leading to tree mortality. So far, EAB is responsible for the death and decline of tens of millions of trees across North America. And these infestations are just widespread. It's found currently in 36 states and the District of Columbia and some provinces in Canada. All right, in the SLELO PRISM region, emerald ash is currently known to be present in 4 of the 5 counties. And you can see on the map, all the stuff that's colored blue. That is all the areas that we know emerald ash borer is present across New York. So almost all of New York, with a very few exceptions. Particularly, you can see some in the Adirondack region. On that west side, from just west of the Adirondacks, you'll see Lewis County. That is our only county that we have not found emerald ash borer, but that doesn't mean it's not present. That just means that somebody has not bumped into it on the ash trees yet. And I would not be surprised at all if we get a report soon that says emerald ash borer was found in Lewis County. I'm just shocked that I have not, in the last couple of years, heard a report yet. In fact, I ask people that I know live in Lewis County to be on the lookout for it. I have experience with putting in applications for the emerald ash borer biocontrol insects. So I would be, more than happy to assist anybody in that process and and getting them acquainted with how the program works. All right, emerald ash borer management strategies. We use an integrated management approach there to both emerald ash borer management and to invasive species management in general. And those will consist of those involve a chemical, mechanical, and biological control, with the biological control being our long-term strategy for forest management throughout the region, because they're going to continue to do the work. Once they establish themselves, they're going to continue to create a new generation after new generation, and they won't not eradicate the population of emerald ash borer, but what they do is they reduce its population to levels that the ash trees can tolerate. And that's our long-term strategy. They're also, with emerald ash borer, we have an additional strategy that is used for ash trees that's called the mortality monitoring and lingering ash detection. And I'll talk about each of these in the next few slides. All right, starting with chemical treatment. Chemical treatment is generally used on ash trees when EAB is approaching a community as a preventative measure. So it's imminent. It's going to be here any day. It's like if you suspect they're going to be there this year, you can go ahead and start applying the chemicals, because like a trunk injection, so that when they arrive, they're already going to have problems, because they'll be basically feeding they're eating the chemical, which is toxic to them. It's also used when ash trees have low levels of EAB and are still in good health. That's generally less than 30% of the crown thinned. Anything greater than that, it's of lesser effectiveness, let's say. There are several chemicals that can be used as a pesticide for EAB, and the next areas I'm just letting you know right away, I am not a pesticide applicator. It's like I know management strategies, and we have a contract with pesticide applicators. So please then don't get too nitty gritty on the pesticide stuff there. That's the stuff that our contractor knows a little bit more about, but I'll give you general information about what the chemical treatment is all about. Chemicals include imidacloprid, dinotefuran, amamectin, benzoate, and azadirachtin. These pesticides are applied using several methods, including trunk ejection, and I have pictures of a few of those. Bark spray, soil drench, and granules applied to the soil. Only the soil drench of imidacloprid and granules of dinotefuran are available to homeowners. The other insecticides and methods I just mentioned will require a certified pesticide applicator. They are effective at killing EAB, which supports the health of the ash tree, but they're going to require repeat applications, either 1 or 2 years from my understanding on these different chemicals. And so you'll be doing that for forever, basically, if you want those trees to survive. Also, use of these chemicals for large numbers of trees can be costly. Like I said, most of the methods you would have to hire a pesticide applicator to apply, and they don't come cheap. And then you add up the amount of trees that you want. You know, unless you're you know, a wealthy person, it's going to be cost prohibitive. So this is why this method is generally recommended in urban areas or on private properties for individual or small numbers of trees that are of significant value to the community or a landowner. So if you have a single ash tree in your yard and you love that ash tree, and you have the money to keep on doing chemical treatments, then chemical treatments might be right for you. If you own acres of ash trees, I said, you know, that's going to be a whole lot of money that you're going to keep on putting into it. And most people are not going to do that kind of chemical treatment because it's just cost prohibitive. Mechanical treatment. Mechanical treatment involves removing the trees. This is recommended for ash trees that will not be chemically treated for EAB, but they present a safety hazard. So if you're in an urban area with high human activity, or there's a lot of vehicles going down roads, and you have ash trees right next to the road or right next to buildings or railroad tracks or what have you. You know, if you're not planning on doing chemical treatment on those ash trees in those areas, then, you know, the best course of action is to remove them. Oh, also talking about those acres of forest with ash, if you have acres of forest, of ash forest, and they're away from any of that stuff I just mentioned, there's no reason to cut them down. Let them fall down on their own. It's like they're not going to harm anything. In fact, they still will do good in that forest because they provide habitat for wildlife, and they'll return nutrients back to the forest soils. So if you're cutting them down and dragging them out, you're not helping wildlife that way, and you're not resupplying those nutrients that the tree took up to supply nutrients for the next generation of whatever plants or trees are going to be growing next. And talking about that, when the ash trees, whether you're cutting them down or letting them fall down, consideration of what other trees there, you know, could be planted there should be thought out. So, you know, if you know there's that you have a street tree, the ash tree, you cut it down, you know, and say, well, it's like, you know, what to do next. After you take it down, it's like think of another tree there, and one that, you know, doesn't have a invasive species targeting it, and consider buying a tree of that species and putting it in right after you remove the old tree. I mean, same thing could be done in forests. I mean, it really depends on how much regeneration you see going on. But if, you know, that regeneration seems limited, it's like you might want to consider putting seedlings down in the forest over the gaps that the ash trees are forming. All right, another management option is to leave ash trees that are not a safety hazard and monitor their health over time, looking for EAB resilient ash trees. There's a program that uses a strategy. It's called Monitoring and Management Ash, or the MAMA program. It was developed and directed by the Ecological Research Institute. It uses community science to find crucial lingering ash. And what are lingering ash? They're mature, native, untreated ash trees that persist in a healthy condition when all other untreated ash trees have been killed by EAB. And twig samples taken from these trees can be used via a program of grafting and selective breeding to yield trees with increased EAB resistance. The Ecological Research Institute created the following graphs to help people with making their ash tree management decisions. And what it's included here on the right side is using the ash as a to do mortality monitoring and lingering ash detection. And if you see on the left, it's this includes the potential hazard trees that are too costly or impractical to treat with pesticide. And these are reasons to cut them down. And those that are removed to maintain habitat type and other purposes. Other purposes to cut them down is you might want to sell them. So they might be used as lumber. In the center is for those situations where you might want to treat them. And this is what I said before, if it's a special role in a landscape and the treatment is practical and affordable. A third option for the trees that don't need to be cut or treated are not a planted cultivar and not being used for biocontrol release like they are here, is to use them for a mortality monitoring and lingering ash detection. To set up one of these plots, to do the monitoring, you need to set up a monitoring plot. The monitoring plot has a few requirements. One is that you have at least 40 mature ash trees of at least 4 inches in diameter, or DBH. Naturally occurring native ash trees. So basically black, green, white around here, ash that are naturally occurring, meaning they're not planted street kind of trees, and they're not chemically treated for EAB because you can't really tell if they're resistant if you're doing chemical treatments on them. The size of the monitoring plot should be between 1/2 acre and 10 acres, and the shape really does not matter. As long as it's in the 1/2 acre to 10 acres size, shape is irrelevant. It's the monitoring is conducted when the leaves are fully leafed out, but before the autumn leaf drop, so that is somewhere between June and September.
Excuse me, the two that's going in the van, they bring back to campus, we're about to leave. Are you guys taking going back to campus right now? Okay.
Oh, also, it's not on, you don't see it on the slide here, but each one of the ash trees in the monitoring plot, you place tree tags on them, so they all get their own individual number, ID number, and the monitoring will be conducted annually until the ash mortality is over 50%. So however many years that takes, there will be how long you'll do the monitoring. When you do the monitoring each year, you'll collect data, which includes the species of ash that you have, the canopy health class, if there's signs of EAB on the ash tree, what's its diameter breast height or DBH, and the location, so coordinates. All right, if you have an ash stand that meets the requirements and you're interested in setting up a monitoring plot, you can contact the Ecological Research Institute. We have like 4 or 5 ones that we set up a few years back. So, and they welcome more. I mean, the more monitoring plots they have, the better chance that they're going to find EAB resistant ash trees out there, which they can then use to make a whole population of resistant ash trees that they can then reintroduce back into the forests and on the streets in the urban areas. The point of contacts are Jonathan Rosenthal, he's the director, and Dr. Reka Waldova, who's a senior scientist. The email is outreach@monitoringash.org, and their website is monitoringash.org. All right. Last but not least, and really where we're going, our focus is going to continue for the rest of this presentation, is biological control. But I wanted you to know a little bit about the other strategies, so that's why I included them today. The biological control is the use of enemies of the invasive pest to reduce its population. For emerald ash borer, there were four parasitoid wasps that there's four parasitoid wasps that are approved for release in the U.S. Those are Species gallinae, Species agrilli, Cestrasticus planopenisi, and Ubius agrilli. Unfortunately, Species agrilli can't be released up here. It was found in more southern parts of China, and it's just not cold hardy. Let's see. Before the wasps were released, there was research in China and in the United States that revealed the wasps preferred EAB over other insects. So there's no adverse effects that were found erased throughout the environmental assessment. And that's important with every biocontrol. There's a lot of people I talk to that don't really understand biocontrol, the modern biocontrol. They think of the biocontrol like cane toads and stuff like that from decades ago. It is there is a lot of regulation on biocontrol, at least a lot. So so it takes literally years of research in the lab, and to convince the government that when you release these out into the wild, that they will do no harm to the native of any native creature that's out there. That they specifically target the invasive species that they're designed to that they're being released for. These wasps so far have been released in over 350 counties in 30 states that are infested with EAB, and Washington, D.C., and 4 provinces in Canada, which is all done by the USDA APHIS, which I'll talk a little bit more about later on. But it's a lot of releases in a lot of areas. So they got a lot of work for them. Recent research has found that the ash regeneration is currently benefiting from the releases of the introduced parasitoids, and it's causing a 20 to 80% parasitism of the EAB larvae in the ash saplings. Those are 1 to 2 inch diameter, and the young trees that are like 5 to 8 inches. It was a big research thing. You can actually read the paper online. So they are affected. So next, I'm going to start with the first parasitoid wasp of the 3 that were released in the last 2 years in Rice Creek, at Rice Creek Field Station. Ubius agrilli targets the inside of EAB eggs, so they're considered an egg parasitoid. They'll inject their own eggs inside the EAB's eggs. The larvae then emerges inside these eggs, and then it feeds on the EAB embryo and kills the egg. There's at least 2 generations per year, and they overwinter as larvae in the EAB egg, and will then emerge as an adult in the spring. The Ubius agrilli can parasitize up to 60% of EAB eggs laid during the summer in some areas of China, and each Ubius adult parasitizes up to 80 EAB eggs during its lifetime. That's pretty significant, a 1 to 80 ratio there. Species gallinae. Species gallinae is actually the newest of the EAB there that was approved, And unlike the other ones, which were found in China, these were found in like Far East Russia. They target the EAB larvae by paralyzing it and laying about 8 eggs on the outside there of the host. So they're what they call ectoparasitoids. The larvae will emerge, feed, grow, and kill the EAB larvae. There's 1 to 2 generations per year. The species overwinters as larvae or pupae and will emerge during the summertime. And what's interesting about Species gallinae, which was not the case with the Species agrilli, which we can't release here anyway, and the other two, for that matter, the other two parasitoid wasps, is that it has a very long ovipositor, which you can see on the left side there, the little, like almost like a little needle coming off the abdomen, and that's what the parasitoid wasp uses to release its eggs. If you see there how long it is, that length is actually good because it's allowing it to actually parasitize the EAB larvae in larger diameter astres. If you have the ones with the shorter ovipositors that are larval parasitoids, like Tachasticus, its ovipositor is not as long. So this actually helps with trees that are up to 23 inches in diameter. Tachasticus planopenisi, it lays its eggs inside the larvae, so it's an endoparasitoid. 1 EAB larva can produce up to 130 Tachasticus adults. So that's quite the big ratio. And there are several generations that are produced each year. The species of parasitoid wasp overwinters as larvae in the host or as a pre-pupae in the host gallery. And they will pupate and emerge as adults in the spring. These are pictures straight from Rice Creek Field Station. The USDA has several different release methods depending on the life stage of the parasitoid wasp and the species. These are an ubinator, which you can see right there on the left on my backpack, which is for Ubius agrilli pupae, and it's tied to an ashtray. And if you look at it, you see it's basically a modified prescription pill bottle. Somebody got clever here. And what they do is they have a EAB. They have EAB in with some paper. You see the paper in there. The EAB lays its eggs on the paper, and then they bring in Ubius and expose Ubius there to that paper, who then inject their eggs into the eggs of the EAB. And then they place the paper into that bottle and then put a little, like a little mesh screen at the very top of it. And then they just put the cap on so that if they emerge, right, if the adult Ubiuses emerge later on, they don't go flying out the bottle. So each time we put one of these on a tree, we just take the cap off. When they emerge, they'll go very straight through that little mesh screen because they are super tiny. And next we have bolts. The ash bolts are for Tachasticus planopenisi or Species gallinae pupae, which is also tied to a tree. And this is also more interesting stuff where they put the eggs of the EAB. EAB lays its eggs on the bolt. And when the larvae emerge, they go inside the piece of wood, and then they expose the piece of wood to Tachasticus planopenisi or Species gallinae adults, who then lay their eggs in or outside there of the larvae that's within that piece of wood. And cups, which you see a whole bunch of cups. That's the way the USDA sends them to me. Those can be adults of any kind. I personally did not receive any ubius or agrilli adults during the 2 years of releases, but you can get them in that form. And if they're in that form, they'll send them to you in these cups. All right. Each year, the USDA APHIS takes applications for new sites for EAB Biocontrol release. For those not familiar with APHIS, it's a piece of the USDA and it stands for Animal and Plant Health Inspection Service. Its mission is to protect the health and value of American agriculture and natural resources, and includes plant protection and quarantine as one of its priorities. So this is where the EAB Biocontrol fits into their mission. Due to the cost of producing these parasite wasps, sites are carefully selected for the highest probability of establishment. So this wasn't we just put an application and they just send it wherever they want. We had to compete to get this released at Rice Creek Field Station with other locations across over half the country. There are several recommended conditions that the USDA APHIS looks for, but these do not have to, they don't have to be met perfectly. So they can be off a little bit. The recommended conditions include naturally forested areas, woodlots, wooded wetlands, riparian area zones, which is an easy check for Rice Creek Field Station. No harvest or development in the next 5 years, which, they agreed to, that we wouldn't, that none of that would occur, and I don't think it would have occurred anyway. But that's part of the agreement that you have to do to get accepted for the program. Avoid sites with excessive human activity. It does mention here like along roads, trails, or railroad tracks in the picnic areas and so forth. And I know we have trails that run around here, but like I said, it doesn't have to be met perfectly. And I don't know, the human activity is not that bad. I'm sure you've all gone to some trails that are just got lots and lots of people on it. The beauty, I think, of these trails here is they're relatively quiet. They're nice trails to walk on. So, they weren't concerned with the trail. The wooded area has to be at least 40 acres. That does not have to fit perfectly because it could actually be less acres in size. And where it says at least 25% of the trees should be ash, they just look for it to be greater. So say you had 20 acres in size, but you had at least 50% ash, that would work itself the same. They would say, yeah, that's fine. Less acres, but more density is great. Variety of ash size classes from seedling to mature trees. So they're looking for they're looking for an opportunity for that regeneration, I think. So if some of those older trees don't make it, you're still going to have a new generation of ash trees coming up behind them, and those would be the ones with the greatest survival from the EAB Biocontrol, which does take a little time to establish itself and increase its populations before it gets fully effective. Low to moderate EAB population densities. Oh. I'm trying to find out where that note is. No, well, basically, it's like you see it down at the bottom. It's like they're the crown class conditions. They're looking for 1 or 2, and it's a 1 through 5 scale. So one is very healthy, while two is healthy, and it just kind of goes back, it goes down to where 4 or 5 is dying or dead. So one of the things that they're looking for as being a prime site is ones that are have more ash trees in the 1 or 2 range for health condition versus the 4 or 5. You know, if most of your trees are dead or dying, you are not a good candidate for release. Fortunately, Rice Creek Field Station met this wonderfully, so we got approved. In fact, when I first put in the application, I was looking around for places to apply for. And so I put the word out to all our partners, and one of our partners is Oswego County Soil and Water Conservation District. And Joe Cervelati, who's director there, he suggested here as a location. And he actually surveyed for all of these conditions and then reported back to me so that I could fill out the application. We submitted the application in the fall of 2021, and it was selected in 2022. Now, we did have a little bit of an advantage when it came to this site in particular, because the USDA APHIS prioritizes the sites they're going to accept by where their high priorities are first release in state, first release in county. And we certainly weren't the first release in the state, but guess what? We were the first release in Oswego County, which is just awesome. So that was one of the reasons why I put in the application, because it was in Oswego County, and of all the counties that have EAB, it was the only one that had not had a prior release, which really just upped our chances of getting approval. So like I said, we did get approved in 2022. And talking about EAB Biocontrol release sites, there are actually 8 that I looked at on the map that the USDA provides, and they've been releasing since 2019, and they break down by 4 locations in St. Lawrence County, 2 in Oneida County, 1 in Jefferson County, and 1 in Oswego County. And of course, Lewis County doesn't have one yet because they haven't had EAB, and that is a prerequisite. If no EAB, like I said, they target EAB. If the parasitoid wasps don't have EAB, they just die. All right, go into a little bit about the program itself. The USDA's EAB Biocontrol Release Program involves 2 years of releasing the EAB parasitoid wasp, followed by 2 years of conducting establishment surveys. So in 2022 and 2023, we completed the release at Rice Creek Field Station. And the total parasitoid wasp by species released were 2,347 Spathis gallinae, 5,652 Tetrachus planopenisi, and 2,200 Ubius. Yeah, Ubius. Should be Ubius agrilae, but anyway, that's a typo there. Should be Ubius agrilae. This is a grand total of 10,199 EAB biocontrol insects released at this site. So a lot of insects. All right, these parasitoid wasps were shipped to us overnight delivery from the rearing lab in Michigan. Upon receipt, which I received them at my office in Pulaski, we would immediately transport them over to Rice Creek Field Station. At the release site, I would meet up with 1 or more of our partner organizations, and We then disperse them to different sections for each release day. And I would look for trees that had some sign of EAB infestation, but they were in relatively good condition. So conditions 1 or 2. You know, because if you're looking at 4 or 5, especially 5, it's like the EAB is going to move elsewhere. So there's no point in releasing there. The release locations and the information about the which species and life stages were released, along with information about the ash trees that we attached the bolts and ubinators to. And some of that information was the DBH of the tree, crown class, whether it had D-shaped holes, epicormic shoots, woodpecker damage. And this was all recorded in a biocontrol app that the USDA provided me with. All right. Now that we are in year 3, all right, we have 2 sampling methods that we can use for evaluating EAB parasitoid establishment. These are the yellow pan traps and tree felling and debarking. Each has its pros and cons. For Rice Creek Field Station, I plan to use the yellow pan traps. And I'll go over more about the yellow pan traps in the next few slides, since that's what we're going to use. But for now, I'll just say that they're good for collecting the adults of all 3 species. They're inexpensive. I mean, really, they're just, you know, if you go into a dollar store and the little yellow bowls you use for picnics, that's the bowl that we use on these. So, and they're easy to make. I assembled 15 of them last weekend. Tree felling and debarking involves cutting down trees and removing the bark to find the larval parasitoids, those being Tetrascus planopenisi and Spacius gallinae, as the Spacius gallinae as eggs, larvae, and pupae, and also the EAB larvae they are on or in. And this, of course, means killing the tree. But it does allow percentage parasitism to be calculated, which is something you can't do with yellow pan traps. But I didn't want to even ask that question. I think the yellow pan traps are just as suitable to the as a sampling method there as the debarking and felling would be. I don't see the point in cutting all the trees down. And there's a lot of extra stuff I won't go into that would be a lot of more time consuming than the yellow pan traps, which I'm, as I'm sure you know, limited on time because I'm responsible for management across 5 counties. Oh, for Obius agrilli, this method collects EAB eggs and the Obius agrilli adults and eggs. These can be collected from dead or alive trees. So in the case of Obius, there you actually don't have to kill them, but the pan traps work fine. What you do with this for Obius is you use a draw knife that shears off thin layers of the outer bark, and that actually leaves the tree, well, unharmed or relatively unharmed. The tree will be fine. So here's the question of the day. How do yellow pan traps work? As crazy as it sounds, the yellow in yellow pan traps is what makes them work. They work because many species of adult bees and wasps are attracted to the color yellow, including the EAB parasitoid wasps that we released at Rice Creek, and some native naturalized parasitoid wasps. And I've listed a few here. They're also actually EAB larval parasitoids, they've come to find out, but they don't target just EAB, so the impact is low. But it's nice that they're helping out and contributing in some way. I mean, the woodpeckers are too, but we certainly can't use those for EAB biocontrol. But they do, the yellow pan trap does also draw in those. So it's basically the color of the pan trap that's the lure for the trap. The wasp, along with the other insects, they'll fly to the color yellow in there, which is the bowl in the trap, which you can see right there. And because there's liquid in there, there's actually propylene glycol, 20% dilution of propylene glycol and some dish soap. They won't really see that because they're both clear. So they'll just fly towards the yellow in the bowl and they'll hit that liquid. And the dish soap actually weakens the surface tension on the liquid. So instead of them hitting it and being able to stand on it, they'll plunge right in like a person diving. And the propylene glycols, we're seeing that, and the dish soap is going to kill them off there, so they'll drown in that. The pan traps are provided by the USDA APHIS. That is, they provide the yellow bowls, the mesh, the shelf brackets, the screws, the zip ties, and the binder clips. But they don't provide everything. So there's a few things we had to provide, which includes a hot glue gun, a portable drill, the dish soap, the propylene glycol, the sample bags, and the and paint thinners, and of course, some assembly required. All right. As was previously mentioned, the yellow pan traps are deployed at the EAB site the following year after the final parasitoid releases. The timing for the yellow pan trap setup depends on the climate in the area and the adult flight period of each species. Above the 40th parallel, which is where we're at, the USDA recommends that deployment of yellow pan traps occurs from spring through early fall. And that basically corresponds to their recommended total growing degree days, base 50. If you're not familiar with that, I'll just briefly explain that. Growing degree days are, they're based on a different base temperatures. In this case, it's 50. So you take the average temperature of the day, subtract 50 from it, and whatever you got is the growing degree days for that for that day. If it comes out a negative number, you just call it a 0. And each day, starting at the beginning of the year, accumulates. So your first growing degree day comes up like here, 10 degrees. If your next one there comes up for 4 degrees, you have 14 growing degrees for the year. For Spathius gallinae and Tostacus planopenisi, the recommended growing degree days for doing the trapping is between 200 and 2900, and for Ubius agrilae, it is between 720 and 2200. So where are we setting up these traps? We plan to set up these traps on 15 ash trees right around the Rice Creek area, right along the trail. The recommendations the USDA has is placing them on the south, southwest, or west sides, is kind of the warmer side, and along forest edges. So we're going to keep them relatively close to the trail system. There's a lot of multiflora rose out there, for all that don't know that, which is an invasive rose, and so it could be a little rough walking through there. And for my sake and the sake of the volunteers, I'm going to try to pick ash trees in the next week or two that are relatively close to the trail, and we're going to do a little cutting of the rows to kind of make a little pass, so to minimize the amount of risk of scratching yourself on the multiflora rose thorns. For Ubius, USDA recommends trees with fresh woodpecker damage and less than 30% dieback. But for Spathius gallinae and Tostacus planopenisi, they state that the higher dieback of up to 89% is perfectly fine. So what does that mean? What am I planning on doing? Well, I'm going to look for trees that have less than 30% dieback, because that's still perfectly fine for the Spathius and Tostacus and meets what they've seen in Ubius. So it would meet all 3 requirements. And because Ubius is usually found around fresh woodpecker damage, I will also look for fresh woodpecker damage there to go along with the less than 30% dieback. We will be checking each trap once a week. Each visit will involve pouring the contents of the trap through a paint filter. We'll have a squirt bottle with us that we'll use to dislodge insects that get stuck in the bowl. The propylene glycol that's in the pan is actually food grade. So it's actually safe to pour on the ground, and that's put out by the USDA. The open end of the paint filter, once we collect or we pour it through, the insects into the paint filter, we'll fold that up and then we'll place it in a labeled Ziploc bag. The trap will then be refilled with a few drops of dish soap and the 27% propylene glycol. All right, once in the lab, the insects will be removed from the paint thinner and placed on a petri dish. Alcohol will be added to help break up the samples. And then sorting for the EAB parasite wasp will occur using a microscope at the lowest magnification. Any of the insects that appear similar to the EAB parasite wasp, we'll pull those to the side. We'll increase the magnification and we'll take a closer look at those. On hot days, it's recommended by the USDA to collect the filters and samples and then put them in a cooler. So if it's a particularly hot day, I may have a cooler with ice packs just for the transporting. The samples can be stored in a refrigerator if processing occurs within the first couple days. But if it takes longer than that, they have to go to a freezer. We don't really have to worry about that because myself, along with some students and or just volunteers and partners, will be sorting them the same day. So we'll collect them from the traps and then we will sort them in the lab same day, so we don't have to worry about that. All the insects suspected of being an EAB parasite wasp are placed in a vial with 95% ethanol. So we're not keying these out to species. That's one reason why we can use volunteers and folks that aren't entomologists. I am not an entomologist, so it's a lot quicker than trying to key them out. If we look, they look similar enough to the parasite wasp that we released, we will place them in a vial. Those vials will then we'll collect those vials for probably about a month and then send all those vials out the vials will be stored in the refrigerator. We'll send those vials out in an envelope there, pouring off the alcohol because of postal regulations, to a lab, the USDA lab that they have in Massachusetts. They have entomologists that are trained for this. They will do the keying out, and then they'll send us emails back about whether we found lookalikes or actual parasite wasps that that were the same species as the ones we released. Like I said, we'll be doing yellow pan traps this year and again next year. And we'll keep everybody up. I mean, Kamal and Kristen will always get the latest, greatest news if we find something. And I'm sure they'll spread the word to everybody that has an interest in the health of the ash trees at Rice Creek Field Station and beyond that. And that's the beauty of the biocontrol, unlike chemical, is that these wasps will spread. So once their population gets large enough, they're going to leave Rice Creek Field Station in SUNY Oswego, and they're going to be roaming around all of Oswego County and beyond, which is wonderful. And no fuss from our part, no additional funds. Thank you. I appreciate the time that you gave me to talk about this. Are there any questions?
How far will they, I mean, how quickly will they spread? Does it depend on just how much the population of them grows?
Yeah, I think that there's a lot of varied. I think it depends on how fast the population grows and those natural pathways of EAB infested ash trees that happen to be surrounding. So if this was just a lone site of ash trees and there was no ash trees to be found for miles, it's like they're that might be a little bit more problematic for the spread, but you know, I think it varies, so I don't have an exact number for that. Yes,
Compared to what you released and what you kill in the yellow traps, what percentage of those little wasps are?
that's really hard to tell because like that data earlier where they're saying it's like the parasitoid wasps like Ubius there will inject its egg and you know it'll produce several more Ubius adults there, you know, for each egg. So I mean it's
I guess.
it'll be very little. Let's put it that way. I mean, I would say fractions of a percent. It's like there and nothing to be concerned of with.
So the purpose of the traps is to see if they're reproducing and.
Yes.
I guess I missed the purpose of trapping and sending them to.
The trapping there is so that we know that they are establishing a population. Yeah, because I do work with Ipinopulenta, which is for swallowwort, and it has not been very good for us for overwintering. So our establishment survey after the last 5 years or something like that has turned out zero.
Okay.
So it's important to know if they're actually they actually survive the winter and have a growing population.
What percentage of the ash trees at Rice Creek would you say are like at that 4 or 5 level? I mean, do you have any idea of the total?
I haven't, no, because that would just be a pure estimate. I mean, it'd be nice if somebody actually did a survey for it. That's really the best way to give an accurate number is if somebody came through and surveyed all of the trees going through that trailed in section, but it's not something I did. Health, I mean, most of them are relatively good health, but I did notice a significant difference there last year from the year before. So there is that you can see the damage is getting greater by the year, but yeah, they're compared to some other places I've been to, especially if you look areas to the south of us there in New York, they're like decimated. So the health of our ash trees are relatively good compared to those. And lucky for us, these biocontrols have been established and created as a reaction to other states that have had it for many, many years before us. So we're really getting lucky that as EAB is entering our area, that biocontrol has already been is already readily available. So hopefully we'll be able to save a lot more trees than other areas. I imagine Michigan lost a lot of their trees just in the research process that I told you that is required to get the parasitoid wasps approved for release. We're not running into that problem. So I think we're fortunate. Yes.
Yeah, actually, the damage is not that bad. No, we don't have too many of 4 and 5 level.
Right.
And but since 2022, there is increase. Now we can tell them. So there is trees which are just behind the air garden. We put a tape on them if you want to see the damage. It's just like that. But the damage on this side of the property almost indetectable.
Yeah, it can be oddly vary. And thinking back to those lingering ash plots that I was talking about, if there is a certain small percentage of ash trees that have some resistance. So, and it's really hard to tell if EAB didn't really target a certain tree that seems to be healthy, because sometimes you see a really healthy tree next to a tree that is starting to show significant signs of EAB. And, you know, it's hard to tell if that's for some reason EAB just didn't pick that tree or if there's actually resistance going on. So you can see odd variations, like you said, where some areas this hasn't been touched for whatever reason, while other ones there are. But the health is relatively, it's pretty decent here. If you go to the south, I went to one location closer to Rochester, as a possible site for release, and everything was a 4 or 5.
Wow.
Everything, everything, except for there was a few little baby trees there that I just think that the EAB wouldn't target because they just don't have the diameter. But I had to tell the person that was managing that land that there's no point in putting an application in for this because it's not going to meet the requirements of the USDA. On the other hand, Rice Creek, perfect. Perfect. The health of the ash trees here are good enough there that the USDA thinks that it's a sound place to do the releases.
So at what diameter can the wasp no longer penetrate? Like, how would you kind of estimate? Like where the wasp is not going to be effective because he cannot penetrate the bog.
Yeah, they have it. Was it 20-something inches was what they said the Spacious gallinax?
23.
20, yeah.
So how would that be in terms, like I'm 23 right now, like pretty, like about like that sort of. I don't know. Okay.
Yeah, that way. Like basically wide.
Oh, so you're measuring around the tree.
Yeah, there's a whole mathematical trick. If you have forester tape there, it's uses the whole circumference thing into it. It's the whole other thing. But I used actually I actually used like a yard meter stick.
Okay, yeah.
So I just held it up like this, and it's like it'll tell you how far it is across, and that's what I inputted into the app that they provided me.
Thank you, everybody. If you should have questions, you can stay behind and talk to Robert